Hello everyone, I am Mohammed Hamama, and this is your ASCP preparation camp. In this camp, we will go through each topic on the ASCP lecture list. Today, we'll be exploring a fascinating topic, hemoglobin S, the first type of hemoglobinopathies. Hemoglobinopathy, as the name suggests, involves a disease related to the hemoglobin molecule. Did you know that it's the most prevalent form of genetic disorder, affecting approximately 7% of the global population? Astonishing, isn't it? Every year, around 300,000 children are born with some form of inherited hemoglobin disorder. These conditions arise due to genetic mutations in one or more genes responsible for hemoglobin synthesis. The mutated genes can impact either the proteins that form the hemoglobin molecule itself, known as globin or polypeptide chains, or the proteins involved in their synthesis and regulation. These mutations disrupt the delicate process of hemoglobin production. Hemoglobinopathies can be broadly classified into two categories, structural defects and thalassemias. Today, we'll focus on structural defects, often referred to as qualitative hemoglobinopathies. In qualitative hemoglobinopathies, the synthesis of hemoglobin occurs at a normal or nearly normal rate. However, the resulting hemoglobin molecule has an altered amino acid sequence within the globin chains. This change in amino acid sequence leads to structural and functional defects in the hemoglobin molecule. Imagine a puzzle with a few pieces in the wrong place, the overall picture changes, and so does its function. On the other hand, thalassemias belong to the quantitative category. They are characterized by a reduced rate of hemoglobin synthesis without affecting the amino acid sequence of the globin chains. This decreased production of hemoglobin leads to anemia and triggers the production of other unaffected hemoglobins in an attempt to compensate for the deficiency. Now, let's focus on the fascinating world of structural defects, the qualitative hemoglobinopathies. They encompass a range of conditions with altered hemoglobin molecules that impact both their structure and function. While some hematologists use the term hemoglobinopathies exclusively for structural defects, it's essential to recognize the distinction between qualitative and quantitative defects. Structure of globin genes. The structure of our genes determines the types of proteins our bodies produce. When it comes to hemoglobin, 6 different genes come into play. These genes reside on 2 different chromosomes. On chromosome 16, we have the alpha-like genes, consisting of 2 genes, alpha and zeta. On chromosome 11, we find the beta-like genes, comprising 4 genes, beta, gamma, delta, and epsilon. Typically, in each copy of our genetic material, we have one of each globin gene, except for alpha and gamma, which have 2 copies. This means that within each cell nucleus, we typically have 2 globin genes, except for alpha and gamma, where we have 4. Each globin gene provides instructions for building a specific type of globin chain. The alpha globin genes, HbA1 and HbA2, guide the production of alpha globin chains, while the beta globin gene, HbB, is responsible for the beta globin chain. The gamma globin genes, HbG1 and HbG2, encode the gamma globin chains, and the delta globin gene, HbD, controls the production of the delta globin chain. Hemoglobin development. As our hemoglobin develops from the early stages of embryonic life, different types of globin chains come into play. Initially, only one alpha-like gene, zeta, and one beta-beta-like gene, epsilon, are active. These genes produce zeta and epsilon globin chains, which combine to form hemoglobin Gower 1, zeta 2 epsilon 2. As development progresses, the production of alpha and gamma chains begins, giving rise to hemoglobin Gower 2, alpha 2 epsilon 2, and Hb Portland, zeta 2 gamma 2. In later stages of fetal development, the production of zeta and epsilon chains ceases, leaving only alpha and gamma chains. These chains then come together to create hemoglobin F, alpha 2 gamma 2, commonly known as fetal hemoglobin. After birth, there's a shift in the types of globin chains produced. The production of gamma chains gradually decreases over the first 6 months, and beta chain synthesis takes over. This shift leads to the production of hemoglobin A, alpha-2 beta-2, the most prevalent type of hemoglobin found in adults. Recent research has shed light on two genes called BCL11A and KLF1, which play a role in regulating the production of gamma-globin. Mutations in these genes can result in higher levels of fetal hemoglobin F even in adults. Additionally, the remaining globin gene, delta, becomes active around the time of birth but produces delta chains in small amounts. These chains combine with alpha chains to form a type of adult hemoglobin called hemoglobin A2, alpha-2 delta-2. In healthy adults, the majority of hemoglobin is A, 95%, followed by hemoglobin A2, less than 3.5%, hemoglobin F, less than 1% to 2%. Genetic mutations. Genetic mutations have the power to alter the sequence of amino acids in the hemoglobin molecule, consequently impacting its structure and function. The types of mutations observed in hemoglobinopathies include point mutations, deletions, insertions, and fusions involving the adult globin genes, alpha, beta, gamma, and delta. The most common type of mutation is the point mutation, where a single nucleotide is substituted with a different one, resulting in the substitution of one amino acid in the globin chain. Astoundingly, around 1109 known hemoglobin variants are caused by point mutations alone. Deletions and insertions are other types of mutations that can occur in the hemoglobin genes. Deletions involve the removal of nucleotides, while insertions add extra nucleotides. These mutations often disrupt the reading frame, leading to the loss of synthesis of the corresponding globin chain. In hemoglobinopathies, however, the reading frame is often maintained, resulting in the addition or deletion of one or more amino acids in the globin chain. This alteration can significantly impact the structure and function of the hemoglobin molecule. Remarkably, approximately 72 described hemoglobin variants are caused by deletions, insertions, or both. Chain extensions are yet another intriguing mutation. They occur when there is a mutation in the stop codon of a gene, causing the translation process to extend beyond the normal endpoint. This leads to longer globin chains than usual. These extended chains can either be degraded, causing a quantitative defect, or if the extension is not significant enough for degradation, it can result in a qualitative defect known as hemoglobinopathy. When two normal genes break at similar locations and recombine with each other, gene fusions occur. This extraordinary phenomenon creates fusion genes that produce hybrid globin chains during transcription and translation. These fusion chains fold differently from the original chains, impacting the function of the corresponding hemoglobin. Fascinatingly, 9 fusion globin chains have been identified thus far. These diverse genetic mutations in hemoglobin genes contribute to the wide range of hemoglobin variants and hemoglobinopathies observed in individuals. Each mutation carries its own story, adding to the rich tapestry of human biology. Zygosity. Zygosity refers to the relationship between the number of gene mutations and the resulting impact on hemoglobin function. It is influenced by the number of copies of specific globin genes involved in producing the globin chains. In normal adult globin genes, we have 4 copies of the alpha and gamma genes, while the beta and delta genes have 2 copies each. This means that mutations in the alpha and gamma genes would need to occur twice as often as mutations in the beta and delta genes to have an equivalent impact. However, it's important to note that the gamma and delta genes have minimal usage in adults and play a relatively minor role in hemoglobin function. As a result, mutations in these genes have a lesser effect on overall hemoglobin function. On the other hand, the beta genes are crucial in producing the dominant hemoglobin in adults, known as hemoglobin A. Mutations in the beta genes can have a more significant impact on hemoglobin function. This explains why there are more identified variants in the beta chains compared to the alpha chains. A single mutation in a beta gene is more likely to cause a clinical condition compared to a single mutation in an alpha gene. Based on the zygosity of the mutations, we categorize individuals into 2 states, heterozygous and homozygous. When only one beta gene is mutated, it is referred to as heterozygous. In this state, individuals are often described as having a trait rather than a disease. Conversely, when both beta genes are mutated, it is known as homozygous. In the homozygous state, individuals are more likely to experience clinical manifestations of the genetic defect. The terms disease and trait are commonly used to describe the homozygous disease and heterozygous trait states, respectively. Pathophysiology. Pathophysiology encompasses several factors that contribute to the impact of point mutations on hemoglobin function. These factors include the type of amino acid substitution, its location in the globin chain, and the zygosity or genetic makeup of an individual. The charge and size of the substituted amino acid play a vital role in altering how the globin chain folds. Changes in charge influence the interaction with neighboring amino acids, while size determines the bulkiness of the globin chain. These modifications can have profound effects on the tertiary and quaternary structure of hemoglobin, ultimately impacting its function. Additionally, the specific location of the substitution within the globin chain influences the extent of structural alteration and subsequent hemoglobin function. Let's take sickle cell disease as an example, where a single amino acid substitution results in the formation of long hemoglobin crystals. These crystals cause the characteristic crescent moon or sickle cell shape of red blood cells. Zygosity, referring to the genetic makeup of an individual, further contributes to the pathophysiology of hemoglobinopathies. In beta hemoglobinopathies, zygosity determines the severity of the disease. Homozygous beta hemoglobinopathies, where both beta genes are mutated, lead to the variant hemoglobin becoming the dominant type, with a reduced or absent presence of normal hemoglobin. Examples include sickle cell disease, hemoglobin SS, and hemoglobin C disease, hemoglobin CC. On the other hand, heterozygous beta hemoglobinopathies, where one beta gene is mutated and the other is normal, usually exhibit a distribution of around 50% variant hemoglobin and 50% normal hemoglobin. It's important to note that homozygous sickle cell disease, hemoglobin SS, represents a severe form of the disease, requiring lifelong medical intervention. Conversely, heterozygotes, hemoglobin AS, are more common but typically remain asymptomatic. Structural hemoglobins can be grouped based on their effects. Some abnormal hemoglobins result in hemolytic anemia or methemoglobinemia, while others affect oxygen affinity. Interestingly, certain abnormal hemoglobins have no clinical or functional consequences. Most clinical abnormalities are associated with the beta chain, followed by the alpha chain, while involvement of the gamma and delta chains is rare and usually insignificant due to their limited presence in hemoglobin. Among these abnormal hemoglobins, hemoglobin S, responsible for sickle cell disease, stands as the most frequent and severe variant. Understanding the pathophysiology of hemoglobinopathies unravels the intricate mechanisms behind the clinical symptoms observed in individuals. Nomenclature. Have you ever wondered how scientists name these fascinating hemoglobin mutations? Well, hold on tight because we're about to take you on a thrilling adventure through the captivating world of hemoglobin nomenclature. Let's step back in time to the early days of hemoglobin discovery, where scientists used a simple yet captivating system to name different hemoglobin types. The adventure began with letters of the alphabet, where the normal adult hemoglobin was known as hemoglobin A and the fetal hemoglobin as hemoglobin F. But as more mutations surfaced, the alphabet started to run out of letters. As the catalog of hemoglobin mutations expanded, scientists realized they needed more than just the alphabet. Let's discover the evolution of hemoglobin naming. The discoverer's imagination and geographical inspiration collide, giving birth to intriguing common names for these exceptional variants. Now, let's venture into the realm of the scientific designation, where secrets about hemoglobin variants are unveiled. The scientific designation acts as a treasure map, revealing essential details about the variant's chain, the position of the abnormal amino acid, and the nature of the substitution. Within the world of hemoglobin, single capital letters hold hidden stories and special characteristics. Imagine hemoglobins with the same electrophoretic mobility but distinct amino acid substitutions, such as hemoglobin G Philadelphia, Hemoglobin G Copenhagen, and Hemoglobin C Harlem. These names become windows into the uniqueness of each variant. Sickle cell anemia. History. Our journey begins in the year 1670 when peculiar symptoms were observed in a Ghanaian family. Little did they know, this was the first glimpse into the world of sickle cell anemia. Fast forward to 1910, when a brilliant cardiologist named Herrick reported the first documented case of sickle cell anemia in a West Indian student with severe anemia. The puzzle started to take shape. In 1927, a groundbreaking revelation occurred when Hahn and Gillespie made a significant breakthrough. They discovered that red blood cells change shape and form sickles when deprived of oxygen. But here's the amazing part. These cells can actually revert back to their normal shape when oxygenated again. The connection to the hemoglobin molecule was slowly unraveling. In 1946, an unexpected twist unfolded. Researchers noticed that individuals with sickle cell trait, carrying one copy of the abnormal hemoglobin gene, had a lower risk of malaria parasite infection. Interestingly, this protective effect was most prominent during childhood, providing temporary shield until active immunity developed. Nature's defense mechanisms are full of surprises. In 1949, the renowned scientist Linus Pauling shook the scientific world with his electrifying experiments. Through electrophoresis, he observed that hemoglobin S migrated differently than hemoglobin A. This remarkable finding led to the revelation of an amino acid substitution in the globin chain, which played a crucial role in distinguishing between different forms of sickle cell anemia, etiology and pathophysiology. In this segment, we're delving into the intricate world of sickle cell anemia, uncovering the underlying causes and the fascinating changes that occur within the body. Let's dive right in. Sickle cell anemia, or SCA, is triggered by a genetic mutation in the hemoglobin protein. Under normal circumstances, hemoglobin consists of 2 alpha chains and 2 beta chains, forming a structure known as alpha-2 beta-2. However, in individuals with SCA, a specific change occurs in the beta chain at position 6. This change involves the substitution of glutamic acid with valine due to a mutation in nucleotide 17. This seemingly small alteration has significant consequences for the charge and shape of the hemoglobin molecule. The substitution of valine for glutamic acid results in a decrease of +1 units in charge. This change affects the movement of the hemoglobin molecule in an electric field and alters its interactions within red blood cells. So, how does this genetic mutation lead to the characteristic sickle shape of the red blood cells? Let's unravel the process. When fully oxygenated, the hemoglobin S molecules do not have a hydrophobic pocket for valine to bind to. As a result, the molecules remain soluble, and the RBCs maintain their normal shape. However, when deoxygenated, a hydrophobic pocket is formed, allowing valine from adjacent hemoglobin molecules to bind. This binding initiates the formation of polymers, elongating in a helical structure. Within the world of sickle cells, we encounter 2 forms, reversible and irreversible. Let's explore their characteristics and implications. Reversible sickle cells respond to oxygen levels. When fully oxygenated, they circulate as normal disc-shaped cells. However, upon deoxygenation, they undergo polymerization, increased viscosity, and shape distortion. On the other hand, irreversible sickle cells maintain their shape regardless of oxygen levels or the degree of polymerization. The spleen recognizes these abnormal cells and removes them from circulation to prevent vaso-occlusion. But the story doesn't end there. Intracellular changes and altered phospholipid distribution play crucial roles in the pathophysiology of SCA. When RBCs with hemoglobin S are exposed to low oxygen levels, hemoglobin polymerization occurs. This triggers the activation of specific membrane channels and results in the efflux of potassium and chloride ions, causing dehydration and intensifying the sickling process. Additionally, the distribution of phospholipids in the RBC membrane is altered. Aminophospholipids, which are normally primarily on the inner layer, become more randomly distributed, leading to an increased presence on the outer surface. These alterations in phospholipid distribution contribute to a range of complications associated with SCA, such as hemolysis, vaso-occlusive crises, stroke, and acute chest syndrome. The presence of phosphatidylserine, PS, on the outer surface of RBCs enhances their adherence to vascular endothelial cells, triggering vaso-occlusive crises, coagulation activation, and decreased RBC survival. Clinical features. Sickle cell disease, or SCD, is an intriguing condition that can manifest in various ways. Did you know that the severity of symptoms actually varies among different ethnic groups? It's true. For instance, Indian patients often experience a milder form of the disease compared to African individuals. But here's the thing, SCD is not just one homogeneous entity. Oh no! There are over 1,000 known hemoglobin variants, but only certain genotypes cause severe SCD. Let me take you through them. These 8 genotypes are the real troublemakers when it comes to SCD. They're the ones that can cause severe symptoms. But fear not, there are also genotypes that result in moderate or mild disease. Now, let's dive into the nitty-gritty of SCD. The variability in symptoms and genotypes can be attributed to a delicate balance within the cells, the ratio of hemoglobin S to hemoglobin F. This, along with factors influencing blood vessel constriction and cellular activation, plays a crucial role in shaping the clinical picture of SCD. Picture this, babies with SCD typically sail through the first 6 months of life without showing any symptoms. Why, you ask? Well, it's all thanks to the protective effect of hemoglobin F. But once those mutated B chains start replacing the normal gamma chains, things start to change. As hemoglobin S levels increase and hemoglobin F levels decrease, red blood cells become more vulnerable to hemolysis. This leads to progressive hemolytic anemia and, in some cases, enlargement of the spleen. It's quite a journey these cells go through. But wait, there's more. Many individuals with SCD experience recurring episodes of pain known as crises. And trust me, they can be quite the roller coaster ride. We've got vaso-occlusive, aplastic, megaloblastic, sequestration, and chronic hemolytic crises. It's like a whole theme park of challenges for these patients. Vaso-occlusive crisis, or VOC, takes the crown as the most common reason for hospital visits in SCD patients. It's an intense pain episode that can affect various parts of the body, the bones, lungs, liver, spleen, genitals, eyes, central nervous system, and even the urinary tract. What triggers these crises, you ask? Well, they can be caused by a multitude of factors, from acidosis and hypoxia to dehydration, infection, fever, and even exposure to extreme cold. It's a real adventure for these brave warriors battling SCD. Now, let's unravel the mystery behind vaso-occlusion. While the exact cause is not fully understood, we do know that the polymerisation of hemoglobin S and the sickling of red blood cells play a key role. But it doesn't stop there. Other factors like decreased cell flexibility, adherence of sickle cells to endothelial cells, and activation of various cells in the body also contribute to this process. It's a complex interplay, my friends. All these shenanigans can lead to vaso-occlusion in the tiniest blood vessels, causing quite a bit of trouble. Those dense and less deformable cells, with higher concentrations of hemoglobin S, are particularly susceptible to intracellular polymerization and are at higher risk of obstruction. As you can imagine, these vaso-occlusive episodes can wreak havoc on organs, causing cumulative infarcts. For instance, about 8% to 10% of SCD patients develop ulcers or sores on their lower legs. The interaction between sickle cells and blood vessel walls plays a significant role in this process. Adherence of sickle cells to the vessel walls leads to intimal hyperplasia, which slows down blood flow. But here's an interesting twist, patients with hemoglobin SC disease experience less sickling and fewer adherent red blood cells. It's like a slightly different path in this complex maze. The frequency and duration of these painful episodes can vary greatly from person to person. Some may go through several episodes a year, while others may be lucky enough to have none at all. On average, each episode lasts for around 4 to 5 days. It's a roller coaster of pain and resilience. But hold on tight, because there's more to explore. Splenic infarcts can lead to scarring and reduced splenic function. This, in turn, can cause a condition called splenic sequestration, where blood becomes trapped in the spleen. It's a rapid decline in hemoglobin levels, and the body faces yet another challenge. And here's a sobering fact: acute chest syndrome, characterized by fever, chest pain, and pulmonary infiltrates, is a significant cause of death in adults with SCD. We mustn't underestimate the gravity of this condition. Speaking of grave complications, let's talk about pulmonary hypertension. It affects about 1/3 of SCD patients and is a severe and potentially fatal complication. In SCD, there's a deficiency of a molecule called nitric oxide, or NO, which contributes to vasoconstriction and hypertension. Low levels of NO fail to inhibit endothelin 1, leading to additional vasoconstriction. It's a tangled web of biochemical events. Last but not least, bacterial infections pose a major risk to SCD patients, especially in the first few years of life. During this vulnerable period, they are more susceptible to life-threatening infections. Chronic hemolytic anemia in SCD is like a whirlwind, where red blood cells are destroyed at a faster rate, leading to decreased hemoglobin and hematocrit levels. It's a roller coaster of symptoms. Picture this: jaundice, elevated counts of reticulocytes, and a shorter lifespan for these brave blood cells, only 16 to 20 days. It's a race against time. The spleen plays a crucial role in removing sickle cells, perpetuating the chronic anemia. But over time, this continuous battle can lead to autosplenectomy, a fancy term for the spleen bidding farewell. Now, diagnosing chronic hemolysis in SCD patients isn't always a walk in the park. There are other possible causes of jaundice, like hepatitis and gallstones, which can throw us off the track. It's like finding a needle in a haystack. But fear not. We're equipped with knowledge. Hemolysis releases free hemoglobin, causing a disruption in the arginine nitric oxide pathway. This leads to decreased nitric oxide levels, activating endothelial cells and narrowing blood vessels, cue vasoconstriction. But wait, there's more. Renal dysfunction is another consequence of hemolysis in SCD. We can detect early signs through an increased glomerular filtration rate. As things progress, we encounter the presence of microalbuminuria, proteinuria, and elevated levels of BUN and creatinine. But fret not, brave warriors. There's hope on the horizon. The use of angiotensin-converting enzyme inhibitors, or ASAs, has shown promise in reducing proteinuria in SCD patients. We're making strides. Let's talk about megaloblastic episodes. It's like a sudden pause in the production of new red blood cells, erythropoiesis, due to a lack of folate. Luckily, folic acid supplementation comes to the rescue, ensuring this deficiency is rare in SCD patients in the United States. Now, aplastic episodes, bone marrow failure, is the most common life-threatening hematologic complication in SCD. Usually triggered by infections like parvovirus, they throw a curveball our way. The bone marrow fails to compensate for the decreased lifespan of red blood cells, resulting in a significant drop in hematocrit without an increase in reticulocytes. Thankfully, most aplastic episodes are short-lived and don't require specific treatment. But in severe cases, blood transfusions may be necessary to manage the anemia. We'll overcome these hurdles together. Our hearts, quite literally, are also affected. Severe anemia can lead to an enlarged heart, murmurs, and cardiomegaly. The heart works tirelessly to maintain adequate blood flow and oxygenation. But worry not, we'll find ways to support and heal our hearts. As if that weren't enough, impaired blood supply to the head of the femur and humerus can result in avascular necrosis. It's like a superhero losing their powers. But fear not. Physical therapy and surgery can help relieve the pressure within the affected bones. In severe cases, hip and shoulder implants may be necessary. Leg ulcers, a chronic thorn in our side. They're characterized by slow healing, unstable scars, and recurring at the same site, causing chronic pain. But we'll soldier on, finding ways to alleviate the suffering and promote healing. And finally, microstrokes, a tiny but mighty foe. They can cause headaches, poor school performance, decreased IQ, and central nervous system dysfunction. Detecting microstrokes requires a neurological examination followed by imaging techniques like MRI, transcranial Doppler ultrasonography, or magnetic resonance angiography. We'll stay vigilant. Incidence with malaria and glucose 6-phosphate dehydrogenase deficiency. The sickle gene is most commonly found in Central Africa, the Near East, the Mediterranean region, and parts of India. And guess what? These regions also coincide with the occurrence of a severe form of malaria, cerebral falciparum malaria, in young patients. Coincidence? I think not. Malarial parasites are sneaky little creatures that set up camp inside red blood cells, utilizing the oxygen present within them. But their stay comes at a cost. The red blood cells adopt a sickle shape, causing damage. Imagine a misshapen red blood cell trying to navigate through blood vessels. It's like a bumpy ride, with the cells getting stuck along the way. These injured cells become easy prey for white blood cells responsible for clearing out foreign particles. This selective destruction of red blood cells containing parasites actually helps reduce the number of malaria-causing organisms. It's like a strategic sacrifice, giving the immune system more time to develop immunity. Fascinating, isn't it? Now, let's dive into glucose 6-phosphate dehydrogenase G6PD deficiency, a condition commonly seen in individuals with sickle cell disease. Some researchers believe that G6PD deficiency might offer some protection in individuals with sickle cell disease. However, it's important to note that this relationship hasn't been definitively established through research. The plot thickens. Interestingly, individuals with both sickle cell disease and G6PD deficiency may experience more episodes of hemolysis. In the first 42 months of life, sickle cell disease patients with G6PD deficiency have shown lower levels of stable hemoglobin, higher reticulocytes counts, 3 times the number of acute anemia events, and a higher frequency of vaso-occlusive and infectious events compared to matched patients without G6PD deficiency. It's like a storm brewing within their bodies. But let's not jump to conclusions. The increased hemolysis in these cases is more likely caused by the enzyme abnormality of G6PD, particularly when the population of red blood cells shifts to older cells during a condition called a plastic crisis. Laboratory diagnosis. Laboratory diagnosis plays a crucial role in confirming the presence of sickle cell disease, SCD, and assessing various hematological parameters. Let's delve into the details of the laboratory diagnostic methods and their significance in diagnosing SCD. 1. Peripheral blood film examination. SCD is characterized by normocytic, normochromic red blood cells. A peripheral blood film stained with right stain can reveal various abnormalities. The presence of long, curved cells with pointed ends, known as sickle cells, is a characteristic feature of SCD. Other findings may include poikilocytosis, irregularly shaped cells, anisocytosis, varying cell sizes, target cells, nucleated red blood cells, spherocytes, basophilic stippling, Pappenheimer bodies, and Howell-Jolly bodies. 2. Reticulocytes count and red blood cell indices. SCD is a chronic hemolytic anemia, and the reticulocytes count is typically increased, between 10% and 25%, due to the bone marrow's response to compensate for the hemolysis. The red blood cell distribution width, RDW, is increased due to moderate variations in cell size, but the mean cell volume, MCV, is not significantly elevated despite the increased reticulocytes count. 3. Leukocyte and platelet counts. Moderate leukocytosis, an increase in white blood cell count, is commonly observed in SCD. The count may reach 40 to 50 billion cells per liter. Neutrophilia, increase in neutrophils, and a mild shift toward immature granulocytes may be present. Thrombocytosis, characterized by an elevated platelet count, is typically seen in SCD. 4. Bone marrow examination. Bone marrow examination can reveal erythroid hyperplasia, indicating an attempt to compensate for the anemia seen in SCD. This can be observed through polychromasia, increased reticulocytes, and nucleated red blood cells in the peripheral blood. 5. hemoglobin electrophoresis and other techniques. The definitive diagnosis of SCD involves the confirmation of the presence of hemoglobin S using specialized laboratory techniques. Hemoglobin electrophoresis, high-performance liquid chromatography, HPLC, or capillary electrophoresis are commonly employed to confirm the presence of hemoglobin S. In complex cases, additional methods such as isoelectric focusing, tandem mass spectrometry, or DNA analysis may be necessary. 6. Hemoglobin solubility test. The hemoglobin solubility test is a common screening method to detect the presence of hemoglobin S. This test takes advantage of the decreased solubility of deoxygenated hemoglobin S in a solution, leading to turbidity. Blood is mixed with a buffered salt solution containing a reducing agent, sodium hydrosulfite, and a lysing agent, saponin. Deoxygenated hemoglobin S polymerizes in the solution, causing it to become turbid, while solutions containing non-sickling hemoglobins remain clear. It's important to note that false positive results for hemoglobin S can occur due to factors like hyperlipidemia, rare hemoglobinopathies, or adding excessive blood to the test solution. Therefore, confirmatory testing using specialized methods is necessary to establish the diagnosis. Did you know that sometimes, even our little ones can trick us? It turns out, false negative results can occur in infants younger than 6 months and individuals with low hematocrit levels. Sneaky, huh? But wait, there's more. Hemoglobin can sometimes play tricks on us too. Meet some of its mischievous cousins that can yield a positive result on the solubility test. We have hemoglobin C Harlem, Georgetown, hemoglobin C Zygosity, hemoglobin S Memphis, hemoglobin S Travis, hemoglobin S Antilles, hemoglobin S Providence, hemoglobin S Oman, hemoglobin Alexander, and hemoglobin Portalegre. These hemoglobins are the life of the party, with not just one, but 2 amino acid substitutions. Now, pay attention to this one, hemoglobin S Antilles. It's not your average hemoglobin variant. Oh no! It can cause sickling even in the heterozygous state. Talk about being a troublemaker. So, how do we catch these clever hemoglobin variants? That's where the lab superheroes come in. Alkaline hemoglobin electrophoresis to the rescue. Alkaline hemoglobin electrophoresis separates hemoglobin molecules based on their molecular charge. It's like a sorting hat for our blood cells. Traditionally, cellulose acetate was the go-to medium, but now, agarose medium is the star of the show. It provides better results and saves the day. But wait, there's a twist. Some hemoglobins have similar charges and patterns, making them tricky to separate. Fear not, our superheroes have a secret weapon. They switch to an acid pH, causing some hemoglobins to acquire a negative charge and migrate towards the anode, while others become positively charged and migrate towards the cathode. For example, hemoglobin S might dance with hemoglobin D and hemoglobin G in alkaline electrophoresis, but they part ways in acid electrophoresis. It's like a choreographed dance-off. Now, hold on to your lab coats, because we have some high-tech tools entering the scene: high-performance liquid chromatography, HPLC, and capillary electrophoresis. HPLC, the superstar of diagnosis, uses a cation exchange column to separate different hemoglobin types. With just one sample injection, it's fast, accurate, and super efficient. Unlike electrophoresis, HPLC can identify and quantify low levels of hemoglobin A2 and hemoglobin F. It's like having a magnifying glass for those sneaky hemoglobin variants. HPLC is the go-to technique for diagnosing thalassemias. It measures both normal and abnormal hemoglobin levels with precision, helping us distinguish these fascinating blood disorders. But wait, we're not done yet. Capillary electrophoresis steps in as the sleek and stylish alternative to traditional agarose electrophoresis. Capillary electrophoresis separates hemoglobin types based on their charge using an alkaline buffer. It's like a red carpet event for our hemoglobin molecules. With better separation and smaller volumes, capillary electrophoresis is the rock star of the lab, impressing us all. We'll explore some exciting techniques and advancements in the field. First up, we have the capillary system, a semi-automated marvel that can perform parallel testing of up to 8 samples. With computerized result analysis, it streamlines the process and saves precious time. And the best part? Capillary electrophoresis is cost-effective. Each capillary can handle a whopping 3,000 runs. That's efficiency at its finest. In the not-too-distant past, agarose electrophoresis was the go-to technique for identifying hemoglobin variants. But times are changing. Both capillary electrophoresis and HPLC are gaining popularity. They offer improved accuracy and precision, revolutionizing the way we diagnose hemoglobinopathies. Now, let's dive into a more complex technique called isoelectric focusing, IEF. It's like a high-tech dance for our hemoglobin molecules. IEF separates hemoglobin molecules based on their net charge. Even tiny differences in pH can effectively distinguish them. It's a powerful confirmatory technique, albeit complex and expensive. When it comes to neonatal screening for sickle cell disease, we need a multi-system approach. Adapted IEF, HPLC, and reversed phase HPLC work together to differentiate various hemoglobin variants and thalassemias. But let's shift our focus to reliable biomarkers of inflammation. Predicting complications and monitoring treatment efficacy is crucial in managing sickle cell disease. While white blood cell count is a good predictor, erythrocyte sedimentation rate and C-reactive protein, CRP, exhibit high variability. During vaso-occlusive crisis and acute chest syndrome, CRP and SPLA2 levels skyrocket. They serve as potential biomarkers, giving us insights into the patient's condition. But the quest for biomarkers doesn't end there. Researchers are exploring interleukin-6, interleukin-10, protein S, annexin A5, malondialdehyde, MDA, depleted A-tocopherol, and vascular cell adhesion molecule 1, VCAM1. Currently, interleukin-6, interleukin-10, VCAM1, and SPLA2 show the most promise for predicting impending crises. These biomarkers are like a crystal ball, helping us foresee and prepare for challenges ahead. Now, let's talk about sickle cell trait, a condition where individuals have one copy of the abnormal hemoglobin gene. It's generally benign and doesn't cause significant health effects. Have you heard about sickle cell trait before? Do you know much about it? Can you tell me in the comments? Let's begin. Sickle cell trait is found in around 8% of African Americans and also in individuals of Central American, Asian, and Mediterranean descent. Most individuals with sickle cell trait don't experience symptoms or clinical manifestations. However, under extreme hypoxic conditions, such as severe respiratory infections or unpressurized high-altitude flights, complications can arise. In such situations, sickling of red blood cells can occur, leading to complications like splenic infarcts, brain necrosis, rhabdomyolysis, and, rarely, even death. Impaired kidney perfusion may cause an inability to concentrate urine in some individuals with sickle cell trait. There have been reported cases of renal papillary necrosis with hematuria. The connection between strenuous exercise and severe or fatal events in individuals with sickle cell trait is a topic of debate. While adverse events have been documented, opponents argue that these events can also occur in individuals without sickle cell trait. Not all individuals with sickle cell trait experience adverse events, and the link to fatal sickle crisis is not firmly established. However, it's worth noting that military recruits with sickle cell trait have been found to have a significantly higher risk of exercise-related death compared to recruits with normal hemoglobin. Diagnostic tests for sickle cell trait involve examining the peripheral blood film, which typically shows normal red blood cell morphology with a few target cells. The hemoglobin solubility screening test and hemoglobin electrophoresis or HPLC can detect the presence of hemoglobin S and hemoglobin A. In individuals with sickle cell trait, electrophoresis reveals approximately 40% or less hemoglobin S and approximately 60% or more hemoglobin A, with normal or slightly increased hemoglobin A2 levels and hemoglobin F levels within the reference range. Treatment is not required for sickle cell trait, and it doesn't affect an individual's lifespan. However, it's important to be aware of the condition and take precautions in extreme hypoxic situations. Stay up to date with our latest videos by hitting that subscribe button and activating notifications. Be the first to know when we release new and exciting content. Don't keep all this valuable information to yourself, share this video with your friends who might find it interesting and beneficial. If you have any questions or comments, feel free to leave them down below. Until next time, take care and goodbye.